welcome to the deep dive. Our mission today is to unpack this, totally paradigm-shifting 2025 paper from the journal Cell by Wu and colleagues.
Yeah, it's a fascinating one. We're looking at a really fundamental biological paradox here.
Right. It's all about how our immune system manages this razor-thin line between fighting off an infection and accidentally destroying our own tissues in the process.
Exactly. We're basically looking at the fail-safes of inflammation because it's an area of immunology that has, frankly, just puzzled researchers for decades.
Because, inflammation is non-negotiable for survival, right? You get a cut, it gets red and inflamed, you need that.
You absolutely need it. But unchecked systemic inflammation, which is what we see in things like sepsis, is often what actually kills the patient.
Right. The pathogen starts the fire, but the immune system just, burns down the whole house.
Yes, exactly. And the assumption has long been that chemical signaling pathways just sort of peter out over time, or the inhibitory cytokines eventually override the inflammatory ones.
But this Cell paper just throws a massive curveball into all of that thinking. The authors discovered that the overriding switch isn't a chemical timer at all.
No, it's the physical environment of the tissue itself.
Yeah.
Specifically, a radical drop in pH.
Acidity.
Right, acidity. Combined with the shape-shifting protein acting as an internal cellular thermostat.
Which is wild, because let's set the baseline for you listening here. Blood pH is usually locked in this really rigid window, right? Hovering right between 7.35 and 7.45.
Always. But the researchers, they focused on the local microenvironment during an infection. They used a bacterial trigger called LPS.
Lipopolysaccharide.
Yep, LPS.
Right.
Which effectively simulates a really severe systemic infection in mice. And while the blood pH does drop slightly during that kind of severe inflammation, what happens locally is just drastic.
How drastic are we talking?
In inflamed tissues, within lymph nodes, and even inside solid tumors, the local microenvironment becomes intensely acidic. We are talking about a pH plummeting all the way down to 6.5.
Wow. And keep in mind, pH is logarithmic. So a drop from 7.4 to 6.5 is...
It's an enormous shift in proton concentration. Massive.
Which brings up an immediate question, I think. If the tissue environment drops to a pH of 6.5, how is that a controlled biological response?
Right. Wouldn't bathing a macrophage in that much acid just indiscriminately denature its protein?
Exactly. Just melt the cell and cause a total failure.
You would think so. And honestly, that's precisely why this mechanism went undiscovered for so long. Because we've known cells have these surface receptors designed to monitor environmental acidity.
Sort of like little smoke detectors on the outside of the cell.
Perfectly said. Proteins like GPR65 or GPR68 and even transcription factors linked to hypoxia like HIM-A, they sit right on the cell membrane, effectively acting as external sensors.
So the prevailing theory was just that the macrophage detects the acidic environment using those specific surface receptors, right?
Yeah. It smells the acid outside, signals the nucleus, and triggers a controlled shutdown before the acid causes permanent damage. That was the standard hypothesis.
But the researchers did the definitive test, didn't they?
They did. They utilized macrophages from knockout mice. These are mice genetically engineered to completely lack those specific sensors.
So they deleted GPR65, GPR68, HIF1A?
Exactly. They made the cells essentially blind to external acidity through all the known traditional receptor pathways.
But the twist in the paper is that it didn't matter at all. The knockout macrophages still successfully detected the acidic environment.
Yes, they still actively dialed back their inflammatory response. The external sensors were completely irrelevant to the specific failsafe.
The call was coming from inside the house.
Exactly. The regulatory signal wasn't coming from a receptor on the cell membrane. The sensor had to be internal. The acidic environment was somehow penetrating the cellular machinery itself.
Okay, but before we track down that internal sensor, we need to clarify what the acid is actually doing to the macrophage's programming. Because looking at the source material, the acidic pH doesn't just broadly suppress the cell.
No, not at all. It's not a master volume knob turning down all immune functions equally.
Right. And to figure out exactly what was happening, the researchers applied something called a linear deconvolution model to the gene expression data.
Yeah, let's pause on that because the methodology is really crucial to understand here. When a macrophage is hit with LPFs and an acidic pH simultaneously, the resulting data is just incredibly noisy.
Just a massive web of upregulated and downregulated genes.
Exactly. So how do you isolate the specific effect of the acid? A linear deconvolution model essentially allows you to untangle interacting variables mathematically.
So if you know how the cell reacts to LPS alone, and how it reacts to acid alone, the model can filter those out.
Exactly. It filters them out to reveal the interaction effect. It shows how the acid specifically alters or hijacks the LPS response. It isolates the delta.
And when they ran this analysis, the gene responses fell into 3 really distinct categories. Let's walk through those.
Sure. So the first group of genes proved completely insensitive to the pH drop. These are the fundamental frontline antimicrobial defense genes.
Like TNF and NF-kB, the first responders.
Right. Whether the pH is 7.4 or 6.5, they fire at full capacity. Which makes perfect evolutionary sense, you know?
Because you can't compromise your immediate defense against a pathogen just because the local environment changed a bit.
Exactly. The cell maintains its core antimicrobial integrity. But the second category, the pH repressed genes, is where the failsafe really kicks in.
Yeah, these are the heavy artillery, the systemic drivers of inflammation. Genes like IL-6 and MED1.
Right. These are the signaling molecules that summon the massive adaptive immune system. They induce severe swelling, and they cause massive collateral tissue damage.
And the researchers found that the acidic environment sharply and specifically shuts these heavy hitters down.
Completely turns them off. But the third category is what genuinely surprised me when reading this.
The pH synergistic genes.
Yeah.
The surprises.
Yes. The acid doesn't just ignore or suppress them, it literally hyperactivates them. Genes governing antiviral responses and tissue repair, like IFNB1 and ADAM.
They spiked by an average of 20-fold.
That 20-fold increase is staggering, and it highlights a remarkable biological pivot. Look closely at those 2 genes you mentioned, ED1 and ADAM.
Oh, this is such a cool contradiction. So EDN1 promotes vasoconstriction, right? It restricts blood flow, which is a classic early inflammatory response to, isolate an infection.
Right. And ADAM, conversely, promotes vasodilation. It opens the blood vessels up, which is crucial for tissue repair and clearing out cellular debris.
So under normal physiological pH, the infection triggers the constrictor, EDN1. But as the tissue gets inflamed and the pH drops to 6.5,
the acid physically suppresses the constrictor and massively boosts the dilator, ADN-M. It is a fundamental reprogramming of the macrophage's entire operational strategy.
Okay, let's just do a quick mini recap for you listening to synthesize this. So the acidic environment isn't acting like a simple volume knob turning down the whole macrophage.
No, not at all.
It's more like a precise railway switch.
Right.
Reverting the train away from the tissue-destroying systemic inflammation and forcing it onto the tracks of tissue repair and resolution.
That's a perfect analogy. And to understand how that railway switch is actually thrown, the researchers had to look at the internal pH of the macrophage.
Right, using specialized pH-sensitive fluorescent dyes.
Yeah, they tracked the intracellular environment. And they discovered that as the external tissue pH drops, the inside of the macrophage also acidifies.
It settles right around a pH of 6.6.
Exactly. So the cytoplasm itself becomes acidic, which means whatever's throwing that switch has to be a protein sitting inside the cell that physically alters its behavior at exactly pH 6.6.
And finding that specific protein is no small task. You have to sift through the entire proteome.
Yeah, they utilized a massive bioinformatics scan, analyzing over 50,000 mouse proteins. They were searching for a very particular molecular architecture.
They needed a protein that undergoes a massive shift in its electrical charge, precisely at that pH drop. And the scan flagged a protein called BRD4.
Right. Now, if you follow genetics, BRD4 is a very familiar player. It's a classic epigenetic reader. Its primary job is to bind to acetylated histones and facilitate transcription.
But a transcription factor doesn't typically function as an environmental sensor. So how is BRD4 detecting acid?
The secret lies in a specific structural anomaly within BRD4. It contains an intrinsically disordered region, or an IDR.
Right, an IDR. So we usually conceptualize proteins as having these rigid, highly specific 3D structures, like a classic lock and key model.
Exactly. But an IDR defies that completely. It's a sequence of amino acids that lacks a fixed, stable conformation.
I always think of it like a floppy, unstructured piece of wet spaghetti, just kind of loose and shifting shape.
Wet spaghetti is a great way to picture it. And the biochemical composition of that flexible spaghetti chain is the key. The IDR on BRD4 is highly enriched with 3 specific amino acids.
Histidine, proline, and glutamine, the HPQ sequence.
Right. And out of those 3, histidine is the true environmental sensor.
Let's dig into the chemistry of histidine. Why is it uniquely positioned to sense a pH drop?
It comes down to its pKi value. That's the specific pH at which a molecule gains or loses a proton. Histidine is unique among amino acids because its pKi is right around 6.0 to 6.5.
Oh, wow. Right in the sweet spot.
Exactly. In a normal cellular environment at pH 7.4, histidine is largely uncharged. It's neutral. But the exact moment the intracellular pH drops into that 6.5 range, the imidazole ring on the histidine molecule acts like a chemical sponge.
It just suddenly soaks up all those loose protons in the acidic environment.
Yes, it rapidly binds to them.
And because protons carry a positive charge, this normally floppy neutral chain of wet spaghetti is suddenly just bombarded with positive electrical charges.
You've got it. The local acidification physically alters the electrostatic landscape of the BRD4 protein in an instant.
Okay, so a drop in pH gives the flexible region of BRD4 a sudden positive charge. But how does that electrical shift actually translate to shutting down the heavy artillery genes like IL-6 while leaving the frontline defense like TNF alone?
Right, how does it regulate specific transcription? This brings us to the underlying mechanics of phase separation, biomolecular condensates.
Yes. This is arguably one of the most critical discoveries in modern biophysics. For those following the recent shifts in cellular biology, the old model of the nucleus was basically a soup.
Right, just a soup where proteins float around and randomly bump into the DNA they need to read.
But phase separation proves it's much more organized than that. Under healthy, normal pH conditions, BRD4 utilizes that flexible, wet spaghetti IDR to physically link up with another protein called MED1.
Yes. Because of those intrinsically disordered regions, when BRD4 and MED1 reach a critical concentration, they undergo liquid phase separation.
They demix from the surrounding nucleoplasm, kind of like oil separating from water.
Exactly like oil and water. They form these highly concentrated, localized liquid droplets directly on specific regions of the chromatin, transcriptional condensates.
I love comparing these condensates to a flash mob or like an exclusive VIP lounge. They create this isolated, highly concentrated environment that just pulls in RNA polymerase and all the necessary machinery to transcribe genes at massive volumes.
A VIP lounge is perfect. And here's where it connects back to those dangerous inflammatory genes. Genes like IL-6 aren't driven by standard, simple promoters. They are driven by superenhancers.
Right, superenhancers. Let's explain that. They are massive clusters of regulatory DNA elements, but they can be located thousands of base pairs away from the actual gene they control.
Exactly. And because the DNA is linear, to read a superenhancer, the DNA strand literally has to loop back on itself in 3D space.
And the BRD4 condensates, that VIP lounge flash mob, are basically the glue holding that 3D loop together.
Exactly. They bridge the massive gap between the distant superenhancer and the gene promoter. If you don't have the physical liquid droplet there, the DNA loop just falls apart.
The machinery diffuses, and the superenhancer cannot activate the gene.
Which perfectly explains the failsafe. Think about what happens when the intracellular pH drops to 6.6. The histidines on the BRD4 protein absorb protons.
They act like that sponge.
Right, and they become positively charged.
And when you pack a dense cluster of positive charges into a microscopic liquid droplet, fundamental physics takes over. Like charges repel.
Yes, the electrostatic repulsion violently overcomes the weak interactions that hold the condensate together.
The VIP lounge just explodes. It dissolves.
A liquid droplet violently dissolves, and the proteins scatter back into the nucleoplasm. The flash mob scatters.
And without the condensate acting as the glue, the 3D loop of the superenhancer collapses. The heavy inflammatory genes like IL-6 are just instantly disconnected from their activation machinery,
silencing the systemic inflammatory response entirely. Meanwhile, core defense genes like TNF don't rely on massive superenhancers or BRD4 condensates to function.
which is why they keep firing perfectly fine in the acidic environment. They don't need the VIP lounge.
Exactly. And the proof for this is just gorgeous. They didn't rely on circumstantial genetic data. They utilized SDED microscopy.
Stimulated emission depletion microscopy, which is amazing because it bypasses the traditional diffraction limit of light, right? It lets them visualize these condensates in living cells at the nanoscale.
They literally watched the process unfold in real time. Within 10 minutes of exposing the live macrophages to an acidic environment, the glowing BRD4 condensates physically melted away on the imaging monitors.
Wow, just popped like bubble.
Just melted. And when they restored the pH to 7.4, the condensates reformed. The phase separation is highly dynamic and completely reversible.
But the most compelling evidence comes from their bioengineering. because they needed to prove that the histidine protonation was the exclusive trigger dissolving these condensates.
Right, so they engineered a mutant variant of the BRD4 protein. They systematically targeted the flexible region and swapped out 33 of those histidine amino acids.
And they replaced them with alanine.
Exactly. Because alanine lacks that proton-absorbing ring, its charge remains stable regardless of the pH.
So it's basically immune to the acid. And the result was exactly what the physics predict. When they bathed cells containing this mutant BRD4 and the pH 6.5 acid, the condensates never dissolved.
Right, because the alanine couldn't absorb the protons, the protein never gained the positive charge. The repulsion never happened.
And the inflammatory superenhancers just kept firing indefinitely.
They even did the reverse, a gain-of-function experiment using a completely unrelated protein called FUS. FUS is an RNA-binding protein known to form its own condensates, but it is totally insensitive to acid.
Until they glued a sequence of 12 histidines onto it, right?
Exactly. They synthetically grafted a histidine tag onto FUS and magically engineered it to behave just like BRD4. At pH 6.5, the newly added histidines absorbed protons and the FUS condensates shattered.
That is just incredible. It definitively isolates that histidine-rich sequence as this modular, programmable pH sensor. Okay, let's do a quick bullet recap for you listening to cement this whole pathway.
Good day.
Okay, step 1, an infection starts.
Yep.
Step 2, the interior of the macrophage drops to an acidic pH of 6.6. Step 3, the histidines on the floppy BRD4 protein act like sponges, absorbing protons and gaining a positive charge.
Yeah.
Step 4, because like charges repel, that positive charge causes the liquid condensates, the VIP lounges, to violently pop. And step 5, without the condensates, the DNA loops collapse, and the most dangerous inflammatory genes are instantly disconnected.
It is a flawless mechanical sequence,
Yeah.
but to fully understand this biological circuit, we have to answer the originating variable. What initiates the drop in intracellular pH in the first place?
Right, because it's not just passive acid leaking in from the tissue, is it?
No, it's not passive diffusion at all. The macrophage actively drives its own acidification. This was the final piece of the puzzle.
Right, because when the LPS signal hits the macrophage, it obviously triggers the inflammatory cascades, but it simultaneously triggers a massive shift in cellular metabolism.
Yes. The macrophage rapidly upregulates glycolysis. To fight an infection, the cell demands an immediate, massive influx of ATP energy.
So it achieves this by basically burning through glucose using enzymes called hexokinases.
Exactly. And the inevitable metabolic byproduct of that hyperactive glycolysis is the rapid accumulation of lactic acid and free protons within the cytoplasm.
Wait, so the metabolic engine that powers the inflammatory response is actively generating the exact acid that will eventually shut it down.
Exactly. It is the ultimate negative feedback loop. The macrophage effectively sets its own chemical timer the moment it engages the pathogen.
That is brilliant. And what truly makes this a masterclass in genetic regulation is that BRD4 itself actually controls the hexokinases.
Yes. It regulates the transcription of those exact metabolic enzymes. BRD4 turns on the metabolic engine, the engine produces acid, the acid dissolves the BRD4 condensates, and the whole system powers down.
It is a literal thermostat. If it gets too hot, too inflamed, the acid trips the BRD4 switch, breaking the circuit before the tissue is completely destroyed.
It's perfectly self-contained, and they verified this entirely in vivo. They tracked peritoneal macrophages in living mice during a severe bacterial infection.
So they actually watched this happen in a living organism.
They did. They documented the whole thing. The metabolic surge, the corresponding drop in intracellular pH, the dissolution of the BRD4 condensates, and the precise recalibration of the immune response, all happening live to prevent fatal sepsis.
That just fundamentally reshapes how we understand the nucleus. I mean, the DNA isn't sealed off in a vault, just passively waiting for chemical messengers.
Not at all.
The raw biophysics of the cellular environment, the actual accumulation of protons from metabolism, is directly reaching into the genome and physically dismantling the 3D structures required to read it. We zoomed all the way from red, inflamed tissue down into the nucleus of a single cell, watching these microscopic droplets pop just to keep us safe.
It's incredible, and it presents some profound therapeutic implications, particularly in oncology.
Right, because this isn't just cool biology, it's the future of medicine.
Yeah.
Tumors are highly acidic environments.
Extremely acidic. And this research provides a direct mechanical explanation for how tumors suppress the immune system. The tumor is literally weaponizing the failsafe.
because by flooding the local environment with acid, the tumor prematurely triggers the protonation of BRD4 inside the invading macrophages.
Exactly. The condensates shatter, and the immune cells are forced to shut down their inflammatory attack before they can clear the cancer. The tumor hijacks the thermostat.
Wow. But understanding that exact biophysical mechanism, that it relies specifically on histidine protonation disrupting the phase separation, that opens up entirely new avenues for intervention, right?
Absolutely. Think about that mutant BRD4 architecture we discussed, the alanine swap.
Oh, right. If we can utilize that, we could theoretically engineer CAR T cells or specialized macrophages with condensates that are totally immune to acid-induced repulsion.
We could create immune cells that simply refuse to shut down in the acidic tumor microenvironment. Or, on the flip side, we could apply this to autoimmune pathology.
Like rheumatoid arthritis or inflammatory bowel disease, where the inflammatory circuit basically fails to deactivate.
Exactly. If we can design small molecules that selectively target that intrinsically disordered region of BRD4 and basically mimic the electrostatic repulsion of the proton,
We could intentionally shatter those condensates. We could physically disconnect the inflammatory superenhancers without relying on broad-spectrum immunosuppressants that wipe out the whole immune system.
precisely. We are looking at a future where we don't just block receptors, we actively modulate the phase separation physics of the genome itself.
It just leaves you with this profound realization about how deeply integrated our biology is with the physical environment.
It really does.
I mean, if you think about it, if our immune system relies on the raw electrostatic charge of accumulated protons to physically assemble and dissolve these temporary liquid structures on our DNA, what other invisible physical forces are currently dictating your gene expression right now?
It's a huge question.
Right. Could the fluctuating physical pressure within your joints, or even the exact internal thermal shifts during a fever, possess their own intrinsically disordered sensors? Are there entirely undiscovered condensates forming and melting inside your cells based purely on physical tension or temperature gradients? Something to think about the next time you get a fever. The architecture of your DNA is listening to the physics of your body a lot closer than we ever imagined.